Welcome back, everyone. Good afternoon. This is lecture 26, the final lecture of this virology course for 2021. And we are going to talk about therapeutic viruses. In my last quotation, trust science, not scientists, by my colleague Dixon Despommier. That's really it. It's the field, not the people. Today we are going to talk about what I call therapeutic viruses. That is, using them to take care of human diseases. And we're going to talk about 4 areas. First, phage therapy for bacterial infections. Very briefly, not a lot. Then gene therapy, delivering a gene to patients who either lack it or have a mutant version of the gene. We'll talk about using viruses to deliver antigens, viral vaccines. We've talked a little bit about this already. We'll talk a little bit about the vectors, and of course, those are in the news. And then viral oncotherapy, using viruses to get rid of cancers. The key for vector development is infectious viral DNA, the ability to take, for any virus, a DNA copy, the genome, whether it's RNA or DNA, make a DNA copy in a plasmid, and then you can modify that DNA copy, introduce it into cells, and out comes virus. First shown for animal viruses. For poliovirus, this is the work that I did as a postdoctoral fellow in 1981. This has since been done for all viruses, and without this, you cannot manipulate the genome. This is the key discovery, to be able to put genes into viruses, to be able to make vaccines, oncotherapy, and so forth. So let's talk briefly about phage therapy. Bacteriophages, right? Viruses of bacteria. After discovering phages in 1915, Félix Derel said, I'm going to use these to cure infections. So he discovered them by finding that the phages lysed bacteria on plates. So he pursued their use as antibacterials, and he founded this Eliava Institute in 1923, which is still around in Georgia, one of the former Soviet states, and it still does phage therapy. This institute produced phages for antibacterial therapy during World War II. You can find kits that were given to soldiers with vials of phages in them to treat infections. However, in the 1930s, the introduction of antimicrobial drugs completely turned off most phage therapy research, because the antimicrobials were easy to do. And the Eliava continued to do it. But now it's having a renaissance, I suppose you could say, because with increasing resistance to antimicrobials, we have to look elsewhere. And so these have been rediscovered, and together with recombinant DNA, the ability to manipulate bacterial genomes and phage genomes, we're making a lot of progress here. And I just want to give you a few examples of what's been going on. So the idea here, first of all, we use lytic bacteriophages, right? Not all bacteriophages kill the cell. Some of them establish lysogeny, and you don't want that. You need to make sure you're working with a lytic bacteriophage that'll kill the bacterium. And then the idea is it has to be specific, right, for whatever the disease-causing bacterium is. You don't want to interfere with your beneficial flora, so you have to make sure about that. So in practice, you have to identify the pathogenic bacterium, and you have to find a phage that will infect and kill it, and then you do the treatment. So it's a little different from a classic drug, right, where it's made way ahead of time. This is kind of tailored to the patient and makes it a little more difficult for FDA approval and all that. There are some phages that have been approved, though. One of them actually is a product of a phage, a protein called the lysine. The lysine is a protein encoded in many phages that lyses the cell. And you can purify these proteins and add them to bacteria. What they will do is break through the peptidoglycan and the membrane of the bacteria, and out come the contents, of course, and bacteria die. So the phage lysins are being explored for surface decontamination. Say in food preparation areas, you could spray them with this and it would kill any bacteria that are there. A phage called agriphage was approved in 2005 to treat a bacterial disease of tomato canker. And List Shield is a phage of the bacteria Listeria, which is FDA approved for treating meat and poultry to eliminate listeria. Listeria often infects meat products and causes serious human disease. So that was the first approval of such a therapy by the FDA, and that is the first designation of generally regarded as safe, which is very important for a product that's going to be designated for human consumption. And there's a lot of research going on now on phage therapy. There have been some interesting clinical successes, but they're few, and it's early days still. For example, these are 3 different publications, and this one on the upper left, phage treatment of an aortic graft infected with Pseudomonas. So this was a patient that received an aortic graft and it got infected, and it would not respond to antimicrobial therapy. So the surgeons injected some phage into the graft. They had to figure out what phage to use, of course, and they cured this gentleman. That's described in this paper. And this is Paul Turner, who was at Yale University, and he works on phage therapy, and he was our guest on Tweevo 44. Really interesting guy. Told a cool story here. And then this one in Nature Medicine, engineered bacteriophages for treatment of a patient with disseminated drug-resistant mycobacterium. This is very interesting because you may know of this high school program called C-phages, where students go out into the dirt behind their house and they purify mycobacterial phages, and then they sequence them and they can annotate them, they can name them after themselves. And so some of them were used in this therapy of this patient. They were modified further to improve their lytic capacity, and then the patient became resistant at some point. So yes, resistance to the phage can arise, so you have to be prepared. And they made modified phages that overcame they made modified phages that could lyse the resistant bacteria. So yes, resistance is an issue, and you can deal with it. But so the gentleman up here with the aortic graft, he died the day the paper was published. This paper, unfortunately, not related to his graft. This young man here, he died, unfortunately, even though his life was prolonged. And then this is an interesting story here on the right. Development and use of personalized phage-based cocktails to treat a patient with disseminated Acinetobacter infection. So this is a story of a guy and his wife. They were traveling, I think, in Egypt, and he got really sick, didn't recover, and they airlifted him to Germany, I think, and they said, well, he's not responding to therapy. And then his wife, I think, worked for the university at San Diego, and she had read about phage therapy. So she started making phone calls and got him to be treated with phage, which was made in part by the Navy and by Ry Young's laboratory at Texas A&M. The guy recovered. They treated him for 2 weeks intravenously, like 10 to the 11th particles a day of phage, and he woke up and he's fine now. And so we did a TWIV there in Texas with Ry and some of the other people who were involved. Very interesting story. They had to make this phage really quickly to treat this guy. So this is early days. I mean, Ry Young says he gets calls every day, and he says, we're not ready to do this in a big scale. We're just learning what can be done. So that is the future. We're going to be using that, I'm sure. Now we're going to talk about viral vectors for doing the things we discussed at the beginning: vaccines, replacing genes, tumor therapy. And of course, a very well-known viral vector is the adenovirus vector, which we've talked about a lot in this course. These are large DNA-containing viruses, of course. Double-stranded DNA, 36 kb or so long. We know a lot about the molecular biology of this virus, as you have learned in this course. The vectors are good. They have a lot of advantages. They can infect postmitotic cells. That means cells that stop dividing, because they can kick them into dividing, right? Remember? I taught you that. Real life stuff. Fast onset of gene expression. The virus remains episomal. Minimal risk of insertion mutagenesis. I can tell you, I get a question every day from someone who wants to know if the adenovirus vaccines are going to integrate in their genome. No, we don't see that. Big capacity. You could delete most of the viral genes, so you can get up to 37 kb if you need it. It's easy to make concentrated preps. A lot of human serotypes are animal serotypes, right? Chimp ad. So you don't have to worry about immunity. The only problem is once you inject it, you get a good immune response. So you may not be able to use the same vector. And that's why the Sputnik vaccine made in Russia, they used 2 different adenovirus serotypes for the first shot and the second shot. Very smart. So vectors undergo evolution. People make them. They fix them. First generation adenovirus vectors, they deleted the E1 and the E3 genes. E1, you should know now what this does. This is encoding T antigens that antagonize RB and P53, and they turn on the early gene expression, transcriptional activation, but you can take it out and grow the virus in a cell line that provides E1. So the E1 deleted virus will not replicate when it infects a cell. It needs E1 for the early gene transcription. So you can grow up the vectors in the E1 producing cell line and have your gene in it and deliver that to patients, and it will not replicate. It will just go in and be transcribed. You use a different promoter than the E1 dependent promoter to drive your transgene, but the viral genes are not going to be expressed. And you can also delete E3, which encodes immunomodulatory proteins. You don't really care about that because these things are not going to be replicating. So this gives you a little bit of space. And in fact, the E1, E3 deleted adenovirus is used for the J&J vaccine, which is unfortunately having problems. Second generation deletions in E2 and E4. In addition, more space for the transgene. And again, we find that these regions are not needed for what we need to do. And then finally, the last generation is called gutless vectors. All the genes are deleted. So here's the adenogenome at the bottom. You take everything out except the inverted terminal repeats at each end and the packaging sequences, the size sequences in the middle here. And then you can put a transgene in here. You can put a promoter of your choice. And you get a lot of capacity if you need it. For the vaccines, we don't need this, so we don't use them. But for other applications, you may have a large gene you want to deliver, and you can do this. And the way you propagate these, you have to encapsulate them, right? You need a helper virus. So this is even more interesting. This is all based on a fundamental study of adenoviruses. So here's our vector DNA. It's all deleted, and we put our transgene in there. So how do we get that in the capsid? Well, you put it in a cell that's been infected with a helper virus. You don't want the helper virus to be in your vector preps, right? So you do a clever thing. Here's the helper virus genome. It's all there. It's E1 deleted. And so you have to do all of this in E1 producing cells. But in addition, the helper has LOX-P sites in the packaging sequence. LOX-P is the sequence that is cleaved by an endonuclease called CRE. It's the CRE-LOX system. It's actually named after cream cheese and lox, CRE-LOX. And I'm not kidding. So when you put this helper, when you infect cells with the helper, it reproduces, makes a lot of capsid protein, but the packaging sequence is deleted. And so it is not packaged. The only thing that's packaged is your vector. Isn't that brilliant? It's based on fundamental stuff that I've taught you. So you may be thinking back then, why do I have to learn this? This is why. Now you understand how these vectors work. Adeno-associated virus vectors. We use those too. Remember this virus? Single-stranded DNA viruses with the weird inverted terminal repeats, not coding for a lot of protein. There's a rep ORF, which is basically the sort of T antigen-like protein that gets DNA synthesis going. Then there's a capsid open reading frame. Well, these have been really popular for gene therapy applications because you get long-term expression. The virus persists without integrating. And this was a surprise. We didn't realize that. And so what you do here is you delete the genes. You take everything, you take out the rep and the cap. So now you can put a transgene in there. You can put a promoter in there. You leave the inverted terminal repeats on. And what you do is you take the plasmid with your transgene, you put it in cells. Along with that, you put a rep and a cap plasmid. Will provide capsid proteins to package the recombinant AAV. But remember, this is adeno-associated. You need adenovirus as a helper. So you add the helper functions on a plasmid, which include E1A, B, E2, 4, and VA RNA. People studied what functions the virus needed. That's them. Now you can use it to make recombinant AAV and deliver genes. And we have a lot of serotypes too, so if you need boosts, not a problem. Now, what happens when you infect cells with these vectors is that the DNA becomes episomal. sometimes you infect cells, the vector can integrate. Now, AAV can integrate only if the rep proteins are present. But if you have a vector, you're not having any rep. Nevertheless, without rep, these DNAs will still persist. So we thought, oh, they're going to integrate, but it turned out that if you take rep away, they don't integrate. But it persists anyway, and that's a result of recombination among the vector sequences. What happens is when you infect your recombinant AAV, the genome begins to reproduce and it recombines into monomers and concatenamers, and those persist for a long time. They're formed by recombination, these concatenamers, and that's why they persist as sort of like episomes in the cells. And again, this was totally unexpected. So that's a really nice vector system because it persists. How long are we talking? Years. As you'll see, this has been used to treat blindness, and those people are years down the line and they're still seeing. Okay, so time for some questions here. What technology is indispensable for the production of therapeutic viruses? X-ray crystallography, high-throughput genome sequencing, synthesis of infectious DNA copies of viral genomes, plaque assay, immunofluorescence. Most of you got synthesis of infectious DNA copies, which is the right answer. That's just indispensable. Without that, you can't do anything. Sequencing helps, but high throughput, you don't really need that. Plaque assay, you don't need plaque assay. You can quantify infectivity in other ways. It's the synthesis of infectious DNA copies for sure. Because otherwise you can't make the deletions I just talked about. You can't put transgenes in. You can't even get going. Retrovirus vectors are used extensively as well. And as you can guess, this is because the DNAs will integrate. So what you can do here is separate the genes on individual plasmids. You can make a GAG-POL or a capsid plasmid and an envelope plasmid. You put these two plasmids into cells, you get empty particles. There's no genome in there. If you want to put something in the particle, you can then add a third plasmid with your transgene on it and a promoter, of course. And you need to have the packaging sequences, right, to get that packaged. And then you will get a retrovirus containing the foreign gene. So again, this is based on fundamental research that people did, saying, what do we need to make a particle? What do we need to get the genome in it? Now, I want to go into a little more detail here about retrovirus vectors because we use two broad types. We use murine leukemia virus-based vectors. These are mouse retroviruses with simple genomes, right? GAG, POL, and envelope. And also we use lentiviral vectors. Can you imagine the virus that's infected 37 million people? We have turned it around to help us. I find this absolutely stunning that we could do that. And what we do in both cases, we make packaging plasmids, we make envelope plasmids for both of these. And then you put your gene in, you make a transgene plasmid as well. They call them transfer plasmids for both. Again, you have LTRs, you have the packaging sequence, a promoter, and your gene of interest. Down here, you see self-inactivating, and that came about because the original transfer plasmids, if they integrate next to an oncogene, what do you think is going to happen? They're going to cause cancer, and they did in a gene therapy trial that we'll talk about. So they make deletions in the 3' LTR so that it's not going to activate a neighboring gene. So basically, only the minimal LTR-derived sequence is there, and it increases their safety. A big piece of U3 is deleted, and so after reverse transcription, a full-length LTR cannot be reconstituted. That way, those are now safe and they're used. So we learned by mistakes. That happens sometimes. So retrovirus vectors, as I said, based on lentiviruses or other retroviruses. You may ask why we used HIV, because they can infect non-dividing cells. Other retroviruses, like the murine leukemia virus-based vectors, their DNA will not get into the nucleus until the cells undergo mitosis when the nuclear membrane breaks down, then they can get in. But HIV doesn't have that restriction. They can infect non-dividing cells. You have long-term expression because, of course, you get integration. You can get up to 8 kb inserts. There is a possibility for insertional mutagenesis, but now we make these SYN vectors that get around that. And we can also pseudotype these, which mean we put a different envelope protein or a different spike protein in them. And you can use VSV spike, which infects a lot of different cells, for example, or you can use a spike of a virus that is restricted, whatever you need to do. So someone asks, do you transfect the cells with all the plasmids at once? Yes, you put them all together, you mix them. You put your capsid, you put your envelope, and you put your transfer plasmid, all three into cells. And out pops retroviruses. It's just amazingly simple. Remarkable that it works, right? Now, pox viruses are also used as vectors. We, of course, have made vaccines years ago with different pox viruses, and they have a big packaging capacity. They don't persist in the host. They are immunogenic, but they're easy to prepare. And modified vaccinia virus Ankara was originally developed to provide an alternative to the smallpox vaccines, which had a lot of side effects. And this is actually the vaccine that's one of the vaccines that's stockpiled. What was done was to adapt the virus by serial passage in primary chicken cells. And that passage led to the loss of the ability to reproduce in mammalian hosts. But the virus can still get into cells. It just won't reproduce. And has a broad tropism and has efficient expression of viral genes and transgenes. And it was used from 1977 to 1980. It was given as a smallpox vaccines to over 120,000 people in Germany. No serious adverse effects. None of the adverse effects that were seen with the regular smallpox vaccine. And it's part of the strategic national stockpile of the U.S., which, as I've told you before, contains an amount of virus to vaccinate every person in the U.S. in the event of a smallpox release. Can you imagine we can do that and we didn't have a coronavirus vaccine ready? So modified vaccinia virus Ankara was then developed as a vaccine vector. And it's been tested in a number of clinical trials. It can be worked on under BSL-1 conditions. The lowest containment in the lab has large capacity. And it's easy to make industrial scale amounts of this. Besides modified vaccinia Ankara, we also use canary pox viruses. All right, that's a pox virus that infects canaries, yes. And it has the same property. It doesn't reproduce in mammalian cells, but it will deliver the genes very efficiently. So here's how you put genes into MVA. Very clever. You clone the transgene in a shuttle vector where the transgene is flanked by viral sequences. Here, the purple. So there's your transgene in red, and the rest is the plasmid. In purple, viral sequences that will allow this to do homologous recombination into the viral genome. You introduce this plasmid into cells that have been infected with, in this case, MVA, and these viruses recombine like crazy, and out come your desired recombinant. You have to screen for them. You can do plaque verification and genome sequencing and verify that it's there. But that's how we make these, and it's really easy to do that. VSV has also been developed as a vector. We use this to talk about principles of negative strand RNA virus reproduction. It's a virus with a long negative stranded RNA genome that encodes 5 mRNAs. And what we do is we replace the gene for the G, the spike, with your gene of interest. It needs to be a spike of some kind, of course. And typically, if we're making a viral vaccine, we're looking to do a spike. We make that, and we can make recombinant particles. And the way you do that is shown at the bottom here. You have 4 plasmids. You have a plasmid for the N, P, and L proteins. And then you have the rest of the genome, the whole genome on the plasmid with your transgene, where you've substituted it for the G. And the substitution of the G, by the way, attenuates the virus so it doesn't cause any disease in people. You transfect those into cells, 4 plasmids, and then each of these plasmids is driven by a bacteriophage T7 promoter. So then you infect the cells with vaccinia virus encoding T7 RNA polymerase. Each of these is transcribed in the cell to make mRNAs, and out come recombinant VSV. So this is T7 polymerase-driven transcription, which is the same technology that's used to make the mRNA vaccines. Flavivirus vectors are also being used. The flaviviruses are plus-strand RNA viruses that encode a polyprotein, and I showed you earlier how we have used the yellow fever vaccine as a backbone to make a dengue virus vaccine. You replace the structural proteins with the dengue virus structural proteins. And the way you would make this vaccine is you have a plasmid with the backbone of the yellow fever vaccine virus with the dengue virus structural proteins. You do in vitro RNA synthesis. You transfect the RNA into cells, and out come your recombinant viruses. Now, taking this further, insect-specific flaviviruses, and one of them is called Binjari virus. They cannot replicate in vertebrate cells, yet they will enter cells and make a lot of protein. So what you do is you take Binjari virus and you replace the structural proteins with whatever virus you want: dengue, Zika virus, West Nile, yellow fever, Japanese encephalitis virus. And they will all reproduce efficiently in insect cells to give you particles, high titer particles. You then immunize mice with them. You get great protection against challenge, and it's been done for Zika virus. So Binjari is an insect-specific flavy. The idea here is you grow up the vaccine in insect cells, and it will infect mammalian cells, but it will not reproduce. So no chance of making disease. Brilliant. More basic research. Similar approach for alpha viruses, which we talked about, plus strand RNA viruses that encode the reproduction proteins at one end of the genome, and then from a subgenomic mRNA, the structural proteins that encapsulate the genome. And so what you do is you replace structural protein genes with whatever you want to make. Typically, it's got to be a virus related to the alpha virus, like chikungunya, so that you can make a capsid. But what is also being done here is to use insect-specific alpha viruses. And this one is called ILAT, E-I-L-A-T. You replace these structural genes with the chikungunya genes. You grow up the virus in mosquito cells, get high titer. Doesn't reproduce in vertebrate cells, but it will infect them, produce the proteins. You can immunize mice with these, and they're protected against challenge. So both insect-specific flaviviruses and alpha viruses are being explored for a number of vaccines, and you'll see them coming out in the future. And when you inject them into your patients and they ask you what's going on here, you can remember all of this, right? Or else go to your laptop and look it up. And finally, Newcastle disease virus. Newcastle disease virus is a virus of chickens. Did you know it's a pathogen? You have to immunize chickens against this. Otherwise, you will lose your flock. So there are already Newcastle disease virus vaccines. It's a negative-stranded RNA virus. It's a paramyxovirus, like measles virus, encodes a number of mRNAs. And what you do is you can insert a transgene in here. And you do, again, the 4-plasmid transfection with T7 polymerase producing vaccinia virus, and you make virus with your transgene. Now, this is being explored for avian influenza virus, avian H5N1 influenza virus. We have to immunize chickens because they can be infected with that. And so when you put the H5N1, say the hemagglutinin in here, you immunize the chickens not only against H5N1, but also against Newcastle disease virus. So you get 2 for the price of 1. And this vector, you may have seen in the New York Times a couple of weeks ago, big article, Mount Sinai, they're using this to make a SARS-CoV-2 vaccine. You can grow this in eggs, make a lot of it. So here are some licensed vaccines that use viral vectors. Ervebo, the glycoprotein from Zaire Ebola virus, is in a VSV vector. This is used in Africa during the current outbreaks. Dengvaxia is dengue in the yellow fever vaccine vector backbone. And also the same yellow fever vaccine vector backbone has been used to make Japanese encephalitis virus vaccines and a West Nile virus vaccine for horses. And as I told you, the Newcastle disease virus vector is used to immunize chickens against both Newcastle disease and H5 avian influenza virus. Here are some experimental human vaccines that use virus vectors. So the previous ones, those are all approved or licensed. An adenotype 5 vectored HIV, gag, pol, neph gene vaccine. Ad26-ZBOV is another Ebola virus vaccine with a glycoprotein in it. We have MVA vectors with influenza H5HA, MVA with HIV envelope. In fact, the RV144H trial I told you about used canary pox to vector the HIV envelope. We have an MVA spike for the MERS coronavirus in camels. And we have a ton of AAV vaccines in development. Herpes simplex, human papillomavirus, HIV-1, and SARS-CoV-1 was made. Experimental vaccines never got very far. A MERS VSV-based vaccine. And basically everything I've told you and more is being explored for SARS-CoV-2. Adenovirus, vaccinia virus, MVA, VSV, Newcastle disease virus, influenza virus, measles virus, all other vectors we haven't talked about. It is being just exploited for everything, of course. All right, so that's vaccines. And you know now that several SARS-CoV-2 vaccines have been given EUAs, if not in the U.S., in other countries, like the chimp adeno 26 vectored spike from AstraZeneca and the J&J ad 21 or 26. I don't remember which one. It's a human adenovirus. Both those are having issues with very rare blood clotting disorders, unfortunately. And we'll see what's going on there. I don't know if it has anything to do with the vector. It might, but then there are other adenovirus vectors being used, and we haven't heard anything. There's one in China. There's the Sputnik in Russia. So we'll see if they have similar issues. Let's talk about gene therapy for monogenic diseases. Monogenic disease is one caused by a mutation in one gene. Why do we do this? Because it's easy to treat. You put one vector in with the gene in it. There are over 6,000 different monogenic diseases of humans. In fact, 1 out of 200 live births, the baby has some kind of monogenic disease. So this is amenable to viral therapy, and there are over 1,800 clinical trials to test vectors for treating various diseases. So these are, again, mutations in the genes that can be inherited in either a dominant or a recessive manner. So here is a table of some of the diseases on the left here, and the defect that we know, the gene defect, the incidence, and then the vectors that are being used to test gene therapy. So, for example, SCID, severe combined immunodeficiency. There are two kinds of defects that can lead to that. Adenosine deaminase, or the common cytokine receptor gamma chain, and retroviruses are being used for that. Hemophilia, hemoglobinopathies, kidney disease, blindness. We'll talk a bit about blindness here. X-linked adrenoleukodystrophy, and others. So there's no shortage of these monogenic diseases. So let's talk about what we know and what we've learned. So here's a table or a graph of clinical trials for gene therapy from 1989 to 2018. And you can see the numbers going up. The number is at the top of each bar. There are some years where it goes up and it goes down. It goes down sometimes because we have a problem, and all of them are canceled for a while until we figure out the problem. You'll see some examples of that. But now we have over 200 such gene therapy trials ongoing. And what are they for? Well, on the left are the indications. Most of them are for cancers. And then we have monogenic diseases that we're talking about now, infectious diseases, diseases of other tissues as well. And you can see most of them here for cancers. And on the right, what vectors? Most of them are adenovirus vectors, followed by retroviruses. Even naked DNA can be injected and be an effective vector as well. And then our other vectors here. I'm just wondering if the problems seen with the adenovirus vectored SARS-CoV-2 spike are going to signal a broader problem that might impact this. We don't know yet. It's too soon to tell, because remember, they've been put in more people now than any adenovirus has been put into people. Over, many, many millions of people. Never been done before. So we'll see what happens. There are two general ways that you deliver a gene in a virus vector. So you can do direct delivery. You put your gene into your virus in ways that we've talked about a little bit today, and you simply inject it into the patient. It's not a great way to do it, because if you do, say, intravenous injection, most of the virus will immediately go to the liver. And if you're trying to target the liver, that's great. But if you want to target something else, you may have to inject it directly into that. So not always the best solution. Cell-based delivery is better. And here we are taking stem cells. We're putting them in culture. So you can purify stem cells from people first we did it from bone marrow, but now we know how to get it from blood. You can culture them, and then you can infect them with your therapeutic virus. You grow them up, make sure the the protein is being produced in the cells, and then you reintroduce them into the patient and their stem cells, so they should colonize and give rise to whatever product. If they're hematopoietic stem cells, they can give rise to lymphocytes and so forth, and many other blood types. So you can purify them from the patient and give them back to the same patient, ideally, right? You can also use embryonic stem cells. You can get in a number of ways. You can get them from the patient. You can get them from cell banks, and then you have to have compatibility. Or you can do stem cell nuclear transplants. You can put the nuclei into the cells from the same person. Or, of course, you can take cells from the person and make them pluripotent and then differentiate them into what you want and then infect them. So those are the two ways we do this. Let's explore some of the ways that have been done and how they've worked. So the very early viral gene therapy trials, one of the earliest, not the first, but this is treating a person for cystic fibrosis. And that's a lung disease where you have a deletion in the cystic fibrosis transmembrane conductance regulator gene, which leads to imbalance of fluids in the lungs and bacterial infections and so forth. So in this case, 1993, this was a male with CF. He was homozygous for a mutation. He was given by putting a tube in his lung. You can see here they're doing that. They're looking at it on the screen there. Put the tube right down into the lung and spray in adenovirus. They sprayed in 10 to the 8th PFU of E1, E3 deleted adenovirus with the DNA for the gene inserted into it. So that was produced in cell culture, of course. And it infects the airway epithelium. And here is the result. So this is production of the protein as assessed in cells from the patient. We're looking at the exogenous protein delivered by the ad vector versus endogenous. So good production. And then it goes down with time. These are 30 days or so. And then they gave him another spray, and he did another boost here, and it went down. They gave him the third one, and nothing happened anymore, probably because of immune responses. And this is what we learned. We didn't know if immunity would be an issue, and this trial showed that it was, and so we had to modify. This person, by the way, was not cured of CF. You have to get it into some kind of a stem cell, most likely, to colonize the epithelium to do that. But we learned a lot from these kinds of therapies, and we learned that immunity of the vectors are an issue. Now, unfortunately, big setback in 1999. First person to die in a gene therapy trial was this teenager, Jesse Gelsinger. He had ornithine transcarbamylase deficiency. That's an X-linked disease, which leads to accumulation of metabolites in the blood. People with the severe forms of this have issues with cognitive ability. They die prematurely. He actually had a mild form of the disease, but he and his dad decided to volunteer for this trial at UPenn. So he was given an adenovirus vector carrying the normal OTC gene. And 4 days later, he died. He had massive immune responses against the vector. He had multiple organ failure. This is highly documented online. You can find the whole story documented. What they did wrong, basically, they broke a lot of rules of conduct at UPenn. Their program was stopped, and many other programs were stopped for a long time. And new rules were put into place to prevent this, because it's really a tragedy. He shouldn't have died. This should have been picked up early, that he was reactive. He had antibodies to the vector. They knew this. They should have not given him the vector, and so forth. Anyway, that's just an example of what can happen because we're doing experimental things. And, he wasn't the only one in this trial, but he was the only one that died. Some of the other patients had, flu-like symptoms and so forth. Some of them had transient liver toxicity. None of this fixed any of their issues. We had to go back to the drawing board. Another example is a trial for X-linked severe combined immune deficiency, SCID. In this case, you have a defect in TB and NK cells. You really have no immune response whatsoever. And these are what would have been called the bubble babies, right? They have to live in a shield. And there were 2 trials done in London and Paris giving infants a retrovirus with the normal IL-2RG, common IL-2 receptor gamma chain, which is, these are all actually mutations in the gene that have been found in many different patients that lead to SCID, all different domains of the gene. So what they did is they took out bone marrow stem cells from these patients. They transduce them with a vector. Transduce means infect. Transduction, of course, is when you have a gene in your virus, you're delivering it. And then they put the cells back into the patients. The disease was actually reversed. Years later, 4 out of the 9 infants in Paris, these are infants now, and 1 in London, developed T-cell leukemia. 3 to 6 years. So their SCID was fixed, but then they developed leukemia. So they stopped. They had 27 trials of using these vectors, and they stopped them all. That's one of those dips in that chart that I showed you. So what happened? They found out by sequencing the genome of these patients that the retroviral vector had integrated next to this gene LMO2. What does it stand for? It's a gene involved in lymphoblastic leukemia. And so basically the insertion of the retroviruses either here, that's patient 4 and patient 5, activated this oncogene constitutively. That gave them the leukemia. They were treated with drugs, with chemotherapy, and that reversed their leukemia. So they were fine. And so they lived on, and they had no issues. They were cured of their SCID, and we got rid of leukemia. But because of this, we decided to modify the retrovirus vectors so that the LTR could no longer activate an oncogene like this. Makes sense, right, from what you know. Could have almost predicted it. I suppose we shouldn't have even done this, but it was done. Fortunately, the kids lived, so that's good. So now these are pretty successful. This is another paper, New England Journal, of infants treated with a lentiviral vector for SCID. 8 infants given a bone marrow transplant with this same gene delivered by a lentivirus to their bone marrow stem cells. And after 18 months, their B and T cells were all good. So it works. It's a really good example of how this can work. You modify and go back and it works. X-linked adrenoleukodystrophy is another example of good gene therapy. This is a disease where you have a defect in a transporter called ABCD1, which leads to fatty acid buildup and damage to the myelin nerve sheath. So what they did is they took out, and this is showing this on a scan here. These white areas are extensive damage to the myelin sheath. So they took out, again, their stem cells. They transduced them with a lentiviral vector with the normal gene, reinfused the cells into the patient. The demyelination was resolved. I think this is after on the left and before on the right, and their neurological status stabilized or improved. I put this here because a former hematologist here, Art Banks at Columbia, was involved in this trial. And he actually wrote the blog post that's on my blog. Right now, other examples of success are retinopathies, inherited retinopathies. These are common, untreatable blinding conditions. And they involve single mutations in all the cells that are needed to see, like the retinal pigment, epithelium, the cones and the rods, the nerves. And in these, there can be defects in many different genes. In each of these arrows here, 4 different arrows, genes in different cell types can be defective and lead to blindness of different types. And this is just a table showing you 3 different kinds of vectors, AAV, lentiviral, and adenovirus vectors that have been used to deliver genes that are defective. RPE genes, for example, cone and rod genes, Muller cell genes, etc. Of all these vectors, the AAV has been the most promising. What you do is you put the gene in your AAV vector, as we have just seen. It's actually injected into the retina. So you have to put a needle into the eyeball and put the needle in the retinal pigment epithelium. You deliver the virus and it infects them. This was studied first in dogs. The disease I want to tell you about is Leber congenital amaurosis. It has mutations in the RPE65 gene, so the retinal pigment epithelium, gene number 65. That's a protein needed for photoreceptor function. And they had a dog model for this where they showed a single subretinal injection of the vector with the canine gene completely restored the visual function of the dogs. And at Penn, Catherine High is one of the people who was driving this program. I interviewed her for TWIV 350. Very interesting story. She's a physician who got interested in gene therapy and did this trial. And now she's started a company to commercialize this. So the FDA approved this therapy in December 2017. Here's the article. It's called Luxturna. I like that name. Lux, right? Luxturna. The other name is Voretigene neparvovec, RYZL, for subretinal injection. First gene therapy approved in the U.S. to target mutations in a single gene, in a specific gene. It's a really good story. I like that. Some other successes are shown here. So SCID, we talked about. ADA is another one leading to SCID. The Leber, by the way, if you want it in 2 eyes, it's $900,000. It's expensive. The companies say, how much is your vision worth? Hemophilia, beta thalassemia, lipoprotein lipase. And then there's this one, the most expensive drug ever made. Avexis is AAV9, adeno-associated virus 9, carrying the spinal motor neuron 1 gene to treat biallelic spinal muscular atrophy. $2.25 million for this. What they're trying to do is recover the costs of the clinical trials, of course, which cost hundreds of millions of dollars. Same thing with the Hep C drugs. They're pretty expensive. And that kind of limits it in other countries, right? So I don't think it's great. I think there's got to be a better way, but it is expensive to do this stuff. Okay, let's have another question. Which of these viral vectors are not likely to be compromised by immune memory in humans? Newcastle disease virus, vaccinia virus, herpes simplex virus, adenovirus. How come it's not green? Because nobody went over, we didn't go over 50%. Most of you got the right answer, though. Newcastle disease virus. It's a chicken virus, right? You're not going to have been infected. You're not likely. Okay, if you're a chicken farmer, how many people are likely to be that? Vaccinia, you would be, because some people got vaccinia vaccine before I did, and the military still gets vaccinia virus-based vaccines. Herpes simplex, right? Everybody has herpes, so that's no good. And adenovirus could be, depending on what serotype. If you use a human ad, it could. So the real clear answer is Newcastle disease virus, chicken virus. Let's talk about treating cancers with viruses, viral oncotherapy. And in general, there's 2 approaches. One, some viruses will selectively replicate in human tumors. And these are non-human animal viruses like myxoma virus, the one that was used in Australia to kill the rabbits, right? It's being used for tumor treatment. And Seneca Valley virus, a picornavirus, probably of cows, that turns out to selectively replicate in some tumors. Or you can modify viruses, human viruses, to target and kill tumors. We often want to add immune enhancement. It turns out you can't just kill the entire tumor with the virus. You have to activate the immune system. And, many tumors are metastasized. So if you do a general activation of the immune system, you do better. This is based on some early science. In the mid-20th century, a number of viruses that infect humans were given to cancer patients to assess therapeutic potential. None of these trials would now be considered capable of yielding useful information. No controls, and within a single investigation, the source of the virus and the route of administration could actually vary from patient to patient. Concentration of virus was typically not known, probably was different among the patients. Trial design issues like giving patients serum, saliva, or tissue extracts from people suffering from hepatitis or mumps, collecting samples from people with hepatitis, continued recruitment of patients, even if some of them die. So, we can't do any of this anymore. Look at the years here, right? 40s, 50s, before we had medical consent sorted out. It's the same years where we got HeLa cells, right? And they didn't know about it. Henrietta Lacks's family. Here are different diseases, different cancers, different viruses giving to people and the number of patients. And you can see very bad things happen. Patients got hepatitis. Gee, I wonder why, if you give them a hepatitis virus, why they would get hepatitis, right? This one is very interesting. They gave people West Nile virus, and a bunch of them got encephalitis. It's the first time we realized West Nile could do that. These gave transient results, which were promising and really are the basis for what we're doing today. Cancer cells have many common properties. We call these cancer hallmarks, right? These are listed here. Immortality, sustained growth, resistance to tumor suppressors, apoptosis, etc. These are viruses that we have used to take care of cancers with specific properties, either the wild-type viruses or mutant viruses. And these are selected so that they reproduce more efficiently in cancer cells with particular hallmarks than in normal cells. Right? This is what guides us to figuring out what kind of virus to give a certain patient. So, if the cancer hallmark includes angiogenesis, we can use certain kinds of viruses because they happen to be particularly well adapted to those kinds of cancers. All right, those are cancer hallmarks. The other aspect of this is that when cells get immortalized, there are a lot of epigenetic changes that occur. And a lot of those, over half of those changes, they're epigenetic changes, histone modification and so forth, affect components of interferon pathways. So if you can actually look at the tumor genome database, the NCI maintains a database of sequences of patient tumors. And you can look and see that there are defects in immune response genes. For example, if you look at this database, there are many mutations in the gene encoding JAK1, which is a signal transduction protein involved in the type 1 interferon receptor. Like 20% of the genomes have mutations in that gene. We can look at what kinds of mutations are present in the cancer and design viruses to overcome them. Facilitating survival of cancer cells is how these mutations accumulate. So we then introduce mutations in the viral genomes that would normally counter, say, interferon defense to limit reproduction in normal cells, but not in cancer cells that have defects in the interferon system, right? So different aspects of the interferon system are defective in cancer. So we design the viruses so that they cannot overcome interferon. We don't need to in the cancer cells, and these viruses will be limited in normal cells. So taking advantage of the fact that many cancers have defects in interferon, say, we can design a virus that will be able to reproduce in the tumor cells, but not in normal cells. Can also do tumor targeting, make the virus bind to, say, a tumor marker, a neoantigen. As tumor cells reproduce, they sustain mutations, and they make new antigens that aren't present on normal cells, and we can identify those and alter viral proteins to bind to them, like Measles virus HA. Herpes simplex virus glycoprotein has been engineered to either contain IL-13, which will bind to tumors, or antibodies against proteins that are present on tumors. So target the virus to those tumors. Adenovirus, we can insert domains into the fiber that will help bind to tumors, or even in the hexon. And we can add adapters to mediate the binding of the adenovirus to the tumor. So many ways we can retarget these viruses so they specifically home in on tumors and not all cells. We can also do post-entry targeting, and these are two aspects of that shown here. One is called positive targeting. So we make a virus containing a gene that's going to kill the cell, some kind of toxin, for example, but you don't want it to be produced in a healthy cell. So you use a promoter that's inactive in normal cells, but is active in a tumor cell, and such promoters can readily be found. So that's called positive targeting. You're going to produce the toxin in a tumor cell. Here's an example on the bottom of negative targeting. In this case, you incorporate into the vector genome the target of a microRNA that is present in normal cells but not in tumor cells. Remember, microRNAs are short RNAs that will target their sequence and destroy the mRNA, basically. So in the normal cell, the mRNA is present and it's in your vector. So the vector is inactivated. It doesn't reproduce in normal cells. You've picked the mRNA target, microRNA target, so that it's not present in the tumor cell. And now the virus will replicate and destroy the tumor cell specifically. And this is just a northern blot showing you that you can screen cells for various microRNAs. LET7A is in a lot of cell types, but here this one, MIR124A, is only in spinal cord and brain. So you can use that to tailor it to your tumor. We can also put drug convertases into vectors so that they kill more cells. In other words, you put a gene into the vector that's going to convert a drug to the active form. In this case, acyclovir. You could put a thymidine kinase in your vector. Remember, TK is not present in normal cells, only in herpes-infected cells, but there's no herpes here. But we put TK into our vector, and then we give the patient acyclovir. And if we've targeted the vector to tumors, it's going to make chain termination happen, and the drug will also potentially diffuse to other cells. You can also put, so those are pro-drug convertases. We can put ion transport proteins in, or immunostimulatory factors. We're going to see examples of this in a moment. Myxomavirus is the same virus introduced into Australia to kill European rabbits. It doesn't replicate in any non-rabbit host, and in fact infects many types of human cancer cells, probably because the cells don't make an antiviral response. There are pathways activated related to transformation. And so the idea here is, let's say you have a cancer and we want to give you lethal chemotherapy or radiation to eliminate the cancer, but that's going to kill your bone marrow. So we take your bone marrow cells out and preserve them and give them back to you after the therapy. The only problem is if there's some cancer cells in your bone marrow, you're screwed, right? They're going to give you your cancer back. So what they do is they take the bone marrow and infect them with this myxoma virus, and it will destroy the cancer cells and leave the bone marrow cells intact. And these are a number of different studies in mice where they have tested this. So here, for example, acute myeloid leukemia in a mouse model, they take the cells, the human AML cells, and they infect them with the virus, myxoma virus, and they put them back in mice. 90% of the mice are free of the cancer cells. In this one with multiple myeloma, 100% of the mice are free. And they can even deliver these viruses to the mice and show destruction of the tumor cells, in this case, intratumoral inoculation. So this is being explored as well. Measles virus is being developed as a anti-cancer, the vaccine strain in particular. This is an infectious attenuated vaccine strain. It preferentially replicates in tumors. And the changes that make it a good vaccine turn out to make it unable to antagonize parts of the innate response. So the virus replicates well in tumors. We've put the gene for a symporter. A symporter will exchange ions in the cell. And so then during treatment of patients, you give them gamma-emitting isotopes so you can see the tumor. So the NIS, the sodium iodide symporter, will bring in these gamma-emitting isotopes into the tumor cells because the NIS is delivered by measles virus. So you can actually visualize or image the tumor cells. And you can then also give beta-emitting isotopes that will get in and induce radiation poisoning of the tumor. And so this is one trial that was done with 2 patients with multiple myeloma. This is a disease, of course, of the cells that make antibodies. And in these patients, they have, as you can see, normal plasmacytoma cells, the cells that make antibodies, and they have clonal ones, which are the cancer ones, clonal PCs. And so this is patient 1 pre-measles virus. This is patient 2, no normal cells at all. And this patient also had a lump on her forehead, a tumor, a myeloma tumor there as well. So they infused them 10 to the 11 particles intravenously. And you can see patient 1 post-measles, no clonal PCs. And so the same with patient 2. And this one patient, the lump on her head went away. Look at that. This is pre and post brain skull scan, basically. And you can see it here, 3 different views, and it's gone. So this looked good. And one of the 2 had a complete remission, and the other, the cancer returned, unfortunately. Herpes virus has also been modified. This is the name of it. It's also called TVEC, which I think is easier to pronounce. And what we do, we take herpes simplex virus, we put in the gene for GM-CSF. So now the infected tumors are going to make this chemokine, and that'll stimulate the production of granulocytes and macrophages, which will further attack the tumor cell. It's deleted for ICP34.5 and US11, which turn out to make it replicate in tumors. It's deleted for ICP47. This gene normally inhibits antigen presentation, so now the cells are going to be recognized by CTLs. This went through phase 1, 2, and 3 for melanoma. They inject it right into the tumors, and they had a 16% response versus GM-CSF alone, and it was approved in 2015 for treatment of myeloma. Now, this will extend your life by a few months at best, so it's not a solution, but it's a start, and it tells us ways that we can do this. Vaccinia has also been explored. We put GM-CSF gene into vaccinia virus. We delete the TK gene because it turns out that many tumors have a lot of that, so this will specifically send the virus to tumors. And this is being tested for the ability to reach metastatic tumors after IV delivery, right? So metastatic tumors are a challenge because you can't inject intratumorally. And they treated 23 patients with all kinds of advanced treatment refractory solid tumors of different organs. The company is called Generex, by the way, a call back to Edward Jenner, right? And the virus was able to replicate in tumors in nearly half of the patients. They had labeled it with beta-gal, and you can see here in rectal, endometrial, and colon cancers, the virus was able to seek out the tumor, and half the patients had anti-tumor activity. So there's a proof of concept that you could deliver intravenously one of these therapies. I mentioned giving prodrug convertases into these vectors. There's two that I'm going to tell you about. Thymidine kinase, which will convert ganciclovir to triphosphate, a chain terminator. Cytosine deaminase converts 5-fluorocytosine to 5-FU, and they will stop DNA replication of tumor cells, and they will also get into bystander cells and affect them as well. That's one of the benefits of this. And one of those experimental viruses is called TOCA511. It's a retrovirus where they've put the gene for cytosine deaminase in. And what you do is you inject the virus either right into the tumor or intravenously, along with giving the patients 5-fluorocytosine. So the cytosine deaminase is produced in the tumor. The 5-fluorocytosine is converted to 5-FU, and that causes cessation of DNA synthesis. And this is in phase 1 and 2 for gliomas of the brain, which are really hard, of course, to treat. Even poliovirus has gotten into the act. This is a drug called polio Sabin repo. And what it is the vaccine strain of poliovirus, Sabin, with the internal ribosomal entry site from rhinovirus 2, and this makes it attenuated. It turns out that many tumor cells upregulate the polio receptor, so this will somewhat preferentially target them. It's been in phase 2 in glioma, for glioma in 61 patients, and a month extended survival. So this needs more work, but it's an interesting approach. I just don't know what they're going to do once polio is eradicated, how they can keep using this, because we're not supposed to have any polio viruses around. And the last one I want to tell you about is a drug called riolysin, which is reovirus. Unmodified reovirus is not pathogenic for humans. It turns out it kills cells with an activated RAS pathway. RAS is one of those oncogenes. And it's actually been being assessed for many different tumors, but it's in a phase 3 for head and neck tumors. And this is the company Oncolytics Biotech, one of the first ones to push forward viral oncotherapy. So you can see there are a whole slew of cancer therapies. And I'm saying in about 10 years, we're going to be routinely treating most cancers with oncolytic viruses. I want you to understand that the therapeutic efficacy is not just killing the tumor cells, but it's activation of anti-tumor immune responses, right? So here we have a tumor cell being infected by an oncolytic virus. It's going to release materials, antigens, right, that are going to be picked up by antigen-presenting cells. Remember, they will present them And then T cells will be activated. We'll have all kinds of adaptive responses that will then go back and attack these tumor cells. So the virus is really starting the cell on a pathway to engage the immune response. Which of the following statements about oncolytic viruses is incorrect? Infection by oncolytic viruses leads to destruction of tumor cells only at the site of virus inoculation. Some viruses of non-human animals can reproduce selectively in human tumor cells. Viruses with both DNA and RNA genomes can be developed as oncolytic agents. Various mutations in genomes that confer tumor-selective reproduction eliminate or impair viral gene products that counter host interferon defense. Which one is incorrect? The first one is incorrect. Infection destroys tumors only at the injection sites. It's not correct. As I showed you, you can have virus disseminated. You can activate immune responses that will destroy distant tumors as well. You can certainly get non-human animal viruses to selectively replicate in tumors. You can alter both DNA and RNA genomes to do this. And you can take away the interferon countering genes in a virus so that it will now reproduce selectively in tumors where interferon pathways are defective. CAR T-cell therapy, cancer immunotherapy, right? Where we deliver a membrane protein, basically, which is composed of a single-chain antibody that's targeting a tumor antigen, a neoantigen that we've identified, linked to the T-cell protein that's a stimulatory or activating protein of the T cell. So the idea is you remove T cells from your patient and you deliver this CAR gene via lentivirus vector. So we're using viral vectors to do this, and you put the T cells back in the patient. They produce the chimeric antigen receptor, and they will destroy the T cells. And this is now being expanded to macrophages, CAR-Max, and NK cells, CAR-NK cells. So viruses are essential for all of these cool things that we are doing. And, everything I've told you today is possible because of basic research. All these therapeutic viruses, fundamental advances, not just virology, recombinant DNA, immunology, clinical science, but a lot of the basic science that I've told you about in this course is used to design these vectors. There's just no way you could do it without it. So I always like to say there has to be always a balance between translational research, that is the desire to treat a disease, and basic research. It has to be a balance. You can't just say, I want to cure cancer, I want to cure diabetes. It doesn't work. You have to let people be curious. Let them study what they want, and cool things will happen. Most of the stuff we learned in this course was just by scientists being curious, not because they had a particular goal, figuring out how viruses package nucleic acids and how promoters work and what genes you can take away. And they weren't doing that because they wanted to cure cancer. It was just curiosity. So that's what I want to leave you with. I want to thank you for taking this course. It's been my pleasure and honor to teach you. And the only thing I ask of you is not to forget, 90% of knowledge is knowing where to find it. You know where to look it up. And you're familiar now with virology. You actually know more virology than 99.9% of the world's population. And so don't forget it. Again, be curious. Always want to know how things work. I do a lot of communication, and I always exhort people to be curious. And by the way, if you're interested, I do a bunch of podcasts besides virology, microbiology, parasitism, immunology, and there's our lectures that I post as well. Neuroscience. I do a blog. I'm all over social media. So follow me somewhere and see what I do. Don't forget to be curious and be passionate about what you do.